Okay, welcome to the seed workshop. If you can grab a seat, please. We're just about to commence the workshop. Sorry? Keys with her.
Hello, everybody. Dixon, Director of the Centre for Mine Site Restoration. Do I need that? Oh, Director, of the Centre. Welcome to a whole series of practitioner workshops. Those that came to the trajectory workshop this morning, which was all about understanding monitoring and predicting closure and the rest, I think all had a fabulous time. But I think we're in for a really special event today, which is the native seed-based restoration side. So this is one of our 5 key pillars within the centre, and the others are policy, ecophysiology, trajectory, seeds. So you'll be championed today by Simon. So I was going to give a few introductory comments, and first and foremost, we meet on Wajak Budja, so we acknowledged their continuing association with the land and the waters of this country. Toilets are outside. They're rather peculiar, so I'll just leave that with you. Do read the signs, female and male. We already had one encounter in there, rather odd. And the escape areas. So we've got, you can help yourself to coffee and tea up the back at any time during the session, and there's some sandwiches for those that didn't get all of their munchies before coming along. Now, the title of this really says it to you: how to plan, how much it costs, and how to make it cheaper. On the 5th of June, we will launch the UN Decade of Ecosystem Restoration. This is an extraordinary event, and I sit on one of the key planning commissions of the UN, which is what's called the Best Practice Commission, which is about understanding what the best practice is. And key and centre for almost all of this are the supply chains to deliver effective global restoration. Now, the scale of what they plan is 350 million hectares. That's the equivalent of around 1,400 million tonnes of native seed. That has to be got from somewhere. It's not on the moon, it's not on Mars yet, not ever likely to be. But understanding that supply chain, because seed underpins virtually the entire business of global restoration, is fundamental to that. It's fascinating that when Simon and I are working on the International Network for Seed-Based Restoration, which we established in 2015 in Santa Fe in the US, and that's now an international group under the umbrella of the Society for Ecological Restoration, our first task was, what was the one thing that all of us and the 450 members and large number of partner organisations in INSR, what was the single thing that we felt was really important? And everyone said, well, it'd be nice to have some standards, because there are no standards. Now, in the US, there's some standards. In Germany, there's some standards. The standards are all different. People were adopting agricultural standards and saying that's all you need. Well, it's fine if you're corn, wheat, maize, rice, barley, and all the rest. But what about Stiphelia serratum? Most you don't even know, or Persunia species. And all the wheels fall off. And so what we've done is create a new international standard for native seeds in restoration, which addresses and goes to the core of how those supply chains need to be robustly understood, so that that 1,400 million tonnes of seed, which currently operates at a wastage rate of around 60 to 90%, simply because the systems of delivery and quality assurance have not delivered those outcomes. And Simon's going to take you through that today. Now, that's more important in Australia than ever before. We have the carbon mania, let's say. It's going nuts by the day. We have Woodside producing 10 million tonnes, 1,100 tonnes of CO2 per hour. So in your workshop today, there'll be 2,200 new tonnes of CO2 put out from just their trains. their liquefaction, let alone all the others that happen. They're very large numbers. Woodside need to find 1 million hectares to plant up each year to offset. What are they doing? Trees, trees, trees. Selling out biodiversity. And as of today, the federal government announced that they are now going to have a biodiversity credits system, particularly for farmers. It's very well resourced for farmers to rebuild biodiversity, and they get paid for it because it matters not only for conservation, but it matters for carbon. So what you're about to work through with Simon and the team in the seed group is actually so relevant and so contemporary in Australia. Otherwise, if we don't get this right, we will look at landscapes in 10 years like we look at the blue gum plantations on the south coast. And we see the catastrophes of the plantings in Eastern Australia and around the world that we've made those mistakes. South Africa, who planted Pinus radiata and Pinus pinastra over huge areas, now it's their major employment program with disadvantaged Indigenous people to pull it all out. It's called working for water because they dried up all their water catchments. So Cape Town, the Mediterranean zone is drying out. They're the mistakes. We can't make those mistakes as we enter on the 5th of June into a decade of restoring the planet. We have to do it right because future generations and many unborn depend upon us getting this right at this point in time. So without further ado, I'd like to hand over to Simon, and I think we're in for an exciting day. And at the end, I'll do a wrap-up about the opportunities for anyone here who has a business or an interest about the way they might wish to engage with our broader research partnership to take it to the next step or to enrich their business going forward. Thank you.
Thank you very much, Kingsley. Okay, thank you very much for coming to this workshop. You're here live and the people that are following us virtually. We've got someone from the Eastern State and even someone from America. Hello, Nancy. It's probably midnight there in Boise, Idaho, but it's great to have you here. So, as Kingsley said, in this workshop, we try to break down some of the aspects of the native seed supply chain in Australia, but globally as well. Before I start, I want to introduce our seed team. We've got Shane. They're all dispersed within you. Shane Turner is our seed dormancy specialist and seed guru in general. Hayley, she's going to be helping you as well, as long as Tiana. Michael, where is Michael? There. And Zoe. So if you've got any question, reach out to them and they can help you out through these activities. So when we talk about a native seed supply chain, I always like to put them in the context of restoration and restorative activities, where we want to go from an ecosystem to be restored to a restored ecosystem. And that usually happens through a restoration plan to start with. And then if you decide you're going to use seeds for your restoration, then you probably need a seed sourcing and procurement plan to understand how many seeds you need, what species, and so on. And usually the supply chain starts from getting those seeds. And in Australia, in Western Australia, it happens almost entirely through seed collection from the wild. And then usually just go all the way to the seed use. Sometimes, especially Europe or America, they have a seed production system to multiply those seeds. Then you can go through seed processing, seed quality testing, seed storage, seed dormancy alleviation if necessary. If appropriate, you can also do some seed enhancement, and then you can finally deliver your seed to the ground. And through monitoring and adjusting management, you ultimately, hopefully, are able to restore your ecosystem. The way of presenting the seed supply chain is the point of view of a supplier. What we're trying to do today is to look at it from the other point of view, the point of view of a practitioner, someone that's tried to bring back this ecosystem using seeds. So instead of going through all of these steps sequentially, we're going to look at it through a series of simulations of a restoration scenario. And as though we scientists at times like to pretend that budget and timelines are not important, those are crucial when you try to get your project done, in budget and in time. So those are things that we'll take into consideration in the simulation, the times and especially the budget. So this entire workshop will work around this concept of a fictional restoration project. we'll kind of arbitrarily, I've decided what species are we going to use and set up a target for this species or what we are trying to get. In this example, it's 1 million. Then we calculate the quantity and the cost of seed needed to achieve that, and then we'll run a simulation. On the left-hand side in red is going to be the simulation cost, cost for seed, cost for the seeding operation, and the total cost. And then we run the real simulation, so how this seeding will turn out. In this case, we'll have a total plant successfully established of 500,000, meaning that we have achieved 50% of our restoration goal. And I've put this indicator here as an indicator of efficiency of seeding, which is the cost of plant successfully established at the end. Because remember, it doesn't matter how many kilos of seeds you put in the ground. What matters is how many plants you get after 3 years or 5 years when you do your monitoring. Because putting the cost for a single plant might be too low, like below 1 cent in some simulations, I've decided to use instead the cost of 1,000 established plant as an indicator of efficiency. I also had to work on another index to measure the success of restoration, because this number alone doesn't tell us much. Because 50% means, yeah, we've got that many plants, but it doesn't tell you about the diversity or how each of the various species is performing. So we tried to put together an index based on the total number of plants established for each species to try to measure the diversity for each species, how it's performing. And it's a number that goes from 0 to 100, 0 being terrible, 100 being perfect. I won't go into the mathematical detail about how we developed this, but I'll show you some example. Like right now, you see most of the grasses that succeeded, not much the acacia and eucalyptus. If we got 0 the acacia eucalyptus in all of the grasses, the index is lower. But if you got more evenly distributed, then the index goes higher. So you'll have all of these appearing while we run the simulation, and you have in front of you this booklet. So if you look, starting from page 15, you'll see we start the first simulation, and there's 2 pages for each simulation. So, oops, lost track. Okay, and on the top of the presentation, you also see what page will it be on. So this is what you can see there, for example. So you've got 1.5 pages to write your notes about that specific topic of the native seed supply chain we'll be discussing. And then here, for you to take notes, there's the 2 boxes of the simulation if you want to record those numbers. You don't need to record them if you don't want to. We're not giving grades if you do it correctly or not, so use it if you think it's useful. So a bit about this simulation, how it works. Well, most of it is based on assumption, educated guesses, and something I completely made it up. For example, species composition, I just made it up. Operation cost, I've got some estimate, but there's also needs some work to be improved. And also the variable for success rate. I based it on the literature and some of the data I've collected, but some of them are still invented. Also, you'll see it's a very simplified simulation. Many of the other costs and variables are not considered. For example, I'm not talking about weeds, I'm not talking about soil preparation, I'm not talking about monitoring. These are all extra costs that eventually need to be accounted for. But for this exercise, I tried to limit everything on what can be controlled within the supply chain and the seed delivery. So it's very simplistic, but hopefully it helps you understand how this process works and how all this improvement in the supply chain can ultimately improve the cost efficiency of seeding. But it's not all entirely made up. In the past year, we've collected information on lots of species, and so we can present you data, real data based on seed price, seed size, quality, and germination. And you'll know that I'll be talking about real data because it'll be having this red outline and it'll be in the corner real data. So these data are mostly based on this series of tests that we run on 191 species that we obtain through the collaboration through the Center for Mine Site Restoration from 2 of the mine sites, one from Perth, Hanson, and one from the Midwest. This is the distribution of the species for life form and families that we performed the study on, and I'll show through the presentation some of the real numbers based on this study. Then we got practical activity. So exactly in the center of your book, there's a part that'll describe the practical activity you'll be doing. Tiana will be talking you through the process of how to perform the seed testing. You're going to perform it on your own. There's seeds in the middle of each of your table of a specific species, and you're going to work on your species through that example. And the descriptions are both on the slides and on your booklet. Okay, that said, after all these explainers, we can start the real presentation and our simulation. So let's get a piece of the wheat belt. Doesn't matter where it is, doesn't matter what it is. It's just a piece of land that we need to restore, let's say, for biodiverse carbon. And we have to do it on 2,000 hectares. Kingsley already touched upon this, that we try to do it as good as we can, following the principle for ecological restoration that they were published in the standards a few years ago. And a key element in this principle that we try to base our project on a reference ecosystem. It's something that we aim towards. So the first thing to do is understand which this native reference ecosystem is and get this model to select our species. In this case, we've got these bits of remnant vegetation, hopefully is healthy enough, and we go in there, understand how they work, what's the species composition, and build our species list based on that one. In this case, as I said, oversimplified, and for the workshop, we get to limit the number of species. We just got 8 species, but I want to select them to be representative of different life forms. So we've got shrubs and trees up here, and on the bottom, we've got the forbs, grasses, and annuals. So try to encapsulate a bit all of the life forms that are present in this sort of ecosystem. And as I said earlier, and I keep on repeating, this is an oversimplified simulation. In a real scenario, you probably need 50, 100, or hundreds of species. So let's go through the species quickly. So each of you on your table will have a different species. Eucalyptus leotopoda, I think, is here. Then we've got Melaleuca radula, second table. Acacia cuminata, the third table. It's very simple. You've got very little processing to do. You're lucky. Then you've got Atriplex amnicola, Boronia simosa, table 5. Ptylothus polystachys on table 6. Syngopogon optectus, the grass. And then for Kingsley alone, Podotheca chrysantha. Kingsley, you'll have to do that one on your own. It's an Asteraceae. So these are the species for the simulation. And let's set up our targets. So what do we want to achieve over these 2,000 hectares? We'll set up an expected return of 5 plants per square meter, which means about 50,000 plants per hectare. 5,000 of which will be tree, the mallee and the shrub. So it means about 1 plant every 2 square meter. Much higher the concentration for the forb, grasses, and annual, about 45,000 per hectare, more or less 5 per square meter. Once we set up the target, we try to put together some numbers. So what is the total number of plants? And it's there, 100 million plants that needs to be delivered over the 2,000 hectares. Now the first question is, how many seeds do I need then to try to achieve those number of plants? The first thing to ask is, how big are those seeds? You cannot understand the quantity you need until you understand each seed, how big they are. There's a few tools online. This is one of the best databases globally to get information about native seeds. It's run by the Millennium Seed Bank, and they've got lots of Western Australia species as well, but they don't have all of them. There's a new tool that RIAWA, the Revegetation Industry of Western Australia, just published. It's a database for native seeds here in Western Australia, and that's another great tool and public database that you can use to get this information. RIAWA, you can ask him. He's the one that developed it. So, as I said, we need to get those information. So I got those information from those species, actually because we collect them ourselves. And those information of seed size, or the seed mass, can be presented in 2 ways. One is very intuitive, number of seeds per gram. So you see the smallest seeds like Melaleuca, you've got 12,000 seeds in a gram. The biggest one, like Acacia, you've got 59 seeds per gram. And that's a huge difference. But a better way to present this number is using the 1,000 seed weight per gram. It means in a gram, what is the weight for 1,000 seed? It's exactly the same information, but it's better to use this information because if you got very big seeds, for example, your santalum, well, in a gram you get half a seed, so it's not really useful to use that tool. This metric is usually much better for that reason. So we'll stick for the rest of the simulation for that one. I want to show you a bit of the data that we've collected. This is the distribution of all the 191 species that we tested for size, and we see how they nicely fit, and you've got most of them sitting between 1 and 10 grams per 1,000 seed. And this is where all your different species sit. So Melaleuca super small, Eucalyptus and Podophyca are quite small, but most of them sits in the middle in the 1 to 10 grams range. So now that we know how much the seeds weight, we can calculate how many seeds we need to get that many plants. So we just divide the 1,000 seeds weight per 1,000 and multiply it by the number of plants needed. And we got the weight in grams. It's about 260 kilograms altogether, which means about 130 grams per hectare. And the next step is, where are we going to get these seeds from? 250 kilo is not that much, but we still need to get them. So should we buy them from a supplier? Should we contract a collector to get them? When we talk about seed collection from the wild, though, there's lots of complexities that come in place. For example, you want to harvest the maximum genetic diversity of the population, and that's a bit tricky. You want to collect enough seeds to do your restoration, but not too many that you damage your population of origin. So it means you can harvest usually more than 15–20% of your population. And if you're dealing with annual species that rely entirely on seeds to go from one year to the next, you shouldn't do more than 10%. And avoid to go to the same population multiple years in a row. If seeds maturation is extended, you might need to go to the same place many times during the year to collect your seeds. And all these things are all the factors that make native seeds very expensive, because collecting from the wild is a very hard job. And on top of that is the very little remnant vegetation left, like in our example. Sometimes there's just not enough seed that you can collect following these rules to do your restoration. That means you need to go there year after year after year to get those seeds. And then you also need to store those seeds and store them properly until you got enough that you can do your restoration. So after all of this, I'm going to show you what the cost for these seeds is. We've got these prices from commercial seed suppliers. And we've got an average cost for a batch of seeds of about $2,000 per kilo. Let's look at the numbers. We've collected information about the cost of seeds on 150 species. We couldn't use all the numbers because some price was way too high. It's considered an outlier, so we got it on 131 species, removing the outliers. And we've got an average of about $1,000 per kilo as a price. If we see how we break it down in different life forms and start putting our species in the different groups, we'll see that more or less they all sit within the range for that life form, apart from Boronia simosa, that sits all the way up there at about $10,000 per kilo. So if we remove that one as an outlier, as we've done for the rest of the data set, we see that the average price for the seeds for this example is about $150 per kilo, which is more or less in line with what we found in the rest of the study at about $1,000 per average. So now we know how much our seeds are, and we can finally calculate the total cost of a seed. So we just multiply the kilo by the cost per kilo, and we'll get a total of about $188,000. And now we can move to the next step, seeding. So, how do we throw those seeds into the ground? One of the most common approaches is to do seed broadcasting. Seed broadcasting can be done manually, in small areas, or where you cannot get access with equipment, or mechanically. So mechanically using tractor, bulldozers, or whatever. Given our scenario and flat farmland, in a very large area, we're probably better off using a tractor, so we use mechanical. Seeding operation cost, that's something that we need to take into account into a simulation. So let's say the cost of a tractor plus the cost of a broadcaster will be $200 an hour, counting also the operator. Maybe it's terribly wrong, you will correct me if it's wrong. Yes? We'll get there. I'm doing it step by steps. Now we're starting from somewhere that absolutely knows nothing of seed and just try to, just chuck them out there. We'll get there eventually. So, tractor and broadcaster, we'll say $200 an hour. Speed, about 5 hectares an hour. So it means to have an average of 50 hectares a day. And it means that in 40 days, we'll be able to complete all our seeding. And that'll bring the total cost of seeding to about $80,000. So we can now finally run our first simulation. Simulation cost, seed, 188,000, adding 80,000 for the operation cost. We've got a total of about 267,000 for the whole operation. And now we look at the results. How did we do? Great, we got 1,650,000 plants, which means we achieved 1.6% of our goal. It's not very good. And the cost of 1,000 established plant is $162 per 1,000 plants. If you look at our index, success index, or diversity index, it's not even at 1. And remember, it's a scale from 0 to 100. So not very good. So the next step is to start looking at what has gone wrong. Because right now, we just put as many seeds as we expected plants to return. But it's not necessarily what happened in nature. If our goal is to get 5 plants per square meter, we are now assuming a 100% success rate. Every seed we put into the ground will turn into a plant. Unfortunately, this is not how it works. So if you put 5 seeds in a square meter, we'll need to start taking into consideration all these other variables that might affect seed success. So we've got depth, seed removal, predation, soil contact, water availability, temperature, competition, and so on. There's so many things that happen in the ground from when you put the seeds to when eventually it turns out to be a healthy and established plants. So the first question is, how many seed reach the appropriate microsite? Because we're doing broadcasting, let's say 20% reach the site. So 1 out of 5 seeds is able to go to the right spot. How many seeds will germinate? Let's say 80%. And that cut down a bit more. How many seedlings emerge? And that usually has been described in the literature to be the big bottleneck between germination and emergence. And it's more or less, usually has been observed to be around 40%. And how many of the seedlings that are able to emerge will eventually survive? Let's say 50%. So if we combine all this number together and try to recalculate how many seeds do we need per square meter to get 5 seeds, it means we need 156 seeds per square meter, which is more or less 30 times more of what we originally put in there. So now that we know this, we can move to the broadcasting, but this time we'll increase the number of seeds, the weight of seeds that we put down by 30 times. So let's try to accommodate, to adjust for that, and let's see how it goes. If you want 30 times those seeds, you're going to end up spending almost $6 million. Adding the 80,000 of seeding operation, you got this total, and then you run the simulation. And our result is almost 50 million plants, better than before. And the cost for 1,000 established plant is about 115. So it's a bit more efficient, but our success index is still at 23%. And remember, we are spending now almost $6 million, and I think it's above anybody's budget. So we have to look at the way we deliver seeds. So far, we talked about seed broadcasting, but broadcasting's got limitations. It's imprecise delivery, and usually is done through surface seeding. So you just drop the seeds on the surface. Imprecise delivery means that you might do under or over seeding in different parts of your restoration site. While surface seeding makes all those seeds more vulnerable to predation, removal for wind and water, a very high surface temperature, and therefore desiccation when the seeds start to emerge. And ultimately leads to a massive seed wastage. You see in our simulation, 1 seed out of 5 was able to reach the appropriate microsite. Instead, if we move to precision seeding, we can now control sowing depth and the spatial distribution of seeds in a more efficient way. We can control the sowing rate, it has a fairly good delivery speed, and it allows as well to deliver multi-species mix. So now if we run what we've seen earlier, the success and demographic process, comparing broadcasting to precision seeding, we'll now have 80% of our seeds able to reach an appropriate microsite. Keeping all the other variables the same, it needs that now we need about 40 seeds per square meter. which is just 8 times more than our initial experiment, not 30. We have to adjust the seeding operation cost because we are no longer using the tractor with a broadcaster. We now have more sophisticated equipment with a precision seeder. Let's say the cost of the tractor with the seeder is similar, but it probably goes at much slower speed, 2 hectares an hour. That means that it's going to take about 100 days to finish the entire operation and bring the cost for this operation up to $200,000. So we can now run the simulation for the precision seeding. We adjust the number, the kilo of seeds required, multiplied by 8, the original one, and the total cost now is $1.5 million, much better than 6. Add the operation cost, and we got a total of 1.7 million. Now we run the simulation, and our establishment is slightly better than before, just the way we adjust the numbers, meaning if we achieve 82% of a restoration goal, and the efficiency, it's much better. It drops from $115 down all the way down to $32. And if you look at our index, it's similar. It's still at 24.7%. I mean, we've got similar results, but we just saved ourselves something like $4 million, so it's not a bad outcome. But now let's go a bit deeper. Let's start looking how each species is doing. And we can see one of them, Podophyca, the Asteraceae, is doing fairly well, it's almost there. But if you look at some of the other ones, some of the trees and shrubs, they're doing terribly, especially Boronia. Nothing came up. We've got absolutely no plant of that species. So that raises our questions. Are we sure that the seeds that we bought, we collected, are actually alive? We start from the assumption that everything we got was perfectly fine, we just put it in the ground. But now, is it really true? Are they really alive? So now we're going into the seed quality test, and I'll let Tiana talk to you about how to perform the seed quality test.
Okay, so Simon's told you how many seeds you're going to need in kilograms, but when you actually look at a batch of seeds, you can see that there's a lot of other material included. So how much seed do we actually have? To work that out, we need to perform a seed quality test. First of all, we want to work out seed purity. In this case, we are performing the test to work out the weight percentage of the batch that can be considered pure seed. It's very simple. You just take a representative sample from your batch, you separate the seeds from other materials. So there could be inert materials such as, for example, twigs, leaves, broken seeds as well, and seeds from other plants. To calculate it, you just take the percentage of the weight of the pure seed, in this case 1.8 grams, divide it by the sample weight, 2 grams, times it by 100, and in this batch we have 90% pure seed units. So we're actually going to do this little activity together. On your tables, you've got a box of equipment and seeds. Just take out a representative sample of those seeds. If you turn to the middle of your booklet, I'll give you guys a second. All right, so if you turn to the middle of your booklet, you'll see this little form that's on page 22. On page 21, there's a set of instructions. You want to fill out this section of the form. What you'll do is you'll follow the set of instructions to separate the seed from other material, and there's a set of balances on this side of the room. For most of you, that'll be on your left side. So you'll just want to weigh out the pure seed unit, the inert section of the batch, and other seeds. If you need any help, just ask any one of our presenters today.
Where is the camera? It's definitely the dog. Like for the sifting, etc.
Sorry?
For the sifting, etc. She was saying.
Yeah, it's, next time if we do it again.
Oral. Sorry, I emailed sorry. Yeah, so she says that
That's amazing. Now I can see it. Is that the focus?
Focus is here. That's the magnification.
Yeah, now I can see it easily.
Yeah, this is real bad. Actually, overall the Melaleucas, these are not too bad because at least they got a color difference.
Yes, exactly. So I can almost do it by color.
Some other Melaleucas, exactly the same color.
Oh, really?
And that's like, we'll show you later how we deal with them, but
Okay.
some are super.
Yeah. Okay, well, I won't separate them. I just want to have another look. It's so much easier with a microscope. Wow. Yep. We started planting up on our farm. Only doing stone. This one, maybe our carbon. Part of the section they're doing fertilizer. I really don't want it. Now they did it one. They want to monitor the difference. So I would ask we actually past Carrara. We're in the tire of Canada. So from the reserve, we are going all the way back. So we have. Okay.
Oh, no. Now it works. Because if you stop it for a bit, then you have to click this one again.
Yeah.
Oh, actually, we're right here.
Okay, are we all ready? Almost. Yeah, yeah. Yes, please, yeah. So the seeds are just over there.
So
But
we're using the
Eucalyptus leptopoda. Yeah, yeah. But that's not going to be for a little while. Yeah, because Simon's going to be talking after this, and then we'll come back to this. Oh, thank you. All right, let's get started with the next section. Kingsley? Okay. So the next part of seed quality testing, we know now what the purity of the seed is, but this doesn't mean that all of those seeds are viable. I forgot one section. So now we can calculate the 1,000 pure seed weight with the seeds that we've separated. These pure seed units, we would take a 50 seed weight, 5 samples of seeds, we get the average weight of those seeds, divide it by 50, and then times by 1,000. That gives us the 1,000 pure seed weight. In this case, Acacia acuminata, the average weight of those 5 samples gives us 0.85 grams. Divide that by 50, you get the weight of a single seed, times by 1,000. In this case, Acacia acuminata, the 1,000 seed weight is 17 grams. You can write these down in your booklet. We won't get you to actually weigh them out and do it yourselves, but this is just something to keep in mind for later. I'll give you a moment to do that. So the next stage, testing viability. Not all of the pure seed units that we've separated are going to be viable, so we have to do a seed fill test. This gives us the proportion of the seed batch that can be considered pure live seeds. Traditionally, there are 2 methods of testing this. First of all, the cut test, where you would take approximately 100 or so seeds and cut into each of those to assess the seed fill. Unfortunately, this is quite time-consuming and it destroys the seed. You can't use that seed that you've cut into. There's also the tetrazolium test. Again, this is time-consuming. The seed has to be soaked, first of all, and then the impermeable membrane is cut, and then it's stained using tetrazolium. This is not only time-consuming, it also requires specialized training, and it can take years of experience to understand the staining patterns for different seed types. And again, it destroys the seed. What we suggest instead is X-ray testing. With X-ray testing, you just take an X-ray image of your seed sample, you count all of the seeds that look healthy, and you cross out all of the ones that are either empty, look like they've been predated, or just look unhealthy or desiccated. I'll just show you this sample video. So it's that quick. To calculate it, again, very simple. First, we want to get the proportion of pure viable seed. So we would count out our filled seeds, divide that number by the total number of seeds in the sample. In this case, we get 0.8 pure viable seed. To calculate the percentage of pure live seeds in your batch, you just times the pure viable seed, so in this case 0.8, by the pure seed units, 0.9, and that gives us 72% pure live seeds. So turn again to the page in your booklet with the form. You're going to be filling out this section. There are X-ray images on each of your tables corresponding to the species that you have. And you should have 2 markers per table in your box.
A bit of a disclaimer before you start. It's going to be very tricky for some species. So if you struggle to interpret what's viable or not, ask us and we can help you out.
Just in case anybody was unsure, you want to count all of the images because those are your 5 samples of 50 seeds.
That should work, yeah.
Perfect. Going on to you. It's your turn. All right, I'm going to hand you over to Simon now, and he's going to run the numbers again for you.
So by now you should have, thank you. So by now you should have all this information about your seed batch. So you know what your viability is, you know what your purity is, and you should have a pure live seed number. We've done the same in the lab. And so for these pieces, this is the result we've got. They're probably going to be different from yours because here you're working on a slightly different batch than what we used originally. But that comes to show how the quality can actually vary between seed batches of the same species. It depends where you collect your seeds. It depends how you store your seeds. There's many variables that comes into place for the quality. So these are our official numbers we've got. Some of these are different. For example, for us, Eucalyptus leotopoda was a 14%, but in this table they've calculated 0.7%. So it really varies a lot depending on the batch. Now when we got this information of a pure live seed, we know that most of the seed that we put in the ground were actually not seeds, or were not able to germinate, were not viable. So to get to the 100% success, we need to adjust for these mistakes and get all this number. It's an average of 22% viability, and we need to adjust it to try to get them to 100%. Before I do that, I want to show you how this number compares to the quality of all the 191 batches that we tested. So for us, the average is 22%. This is the average across all the species, about 56% in total, and that's how it looks among the various species. Grasses are usually the lowest in viability at about 40%. And the ones we got, our species, are about 22%. So in this example, we've been quite unlucky. We've selected species that tend to have a lower viability than the average of the species that we tested. And that's where our species sits within this distribution. So going back to what I was saying before, we need to adjust each species to compensate for the pure live seed. So we do that by dividing 100 by the pure live seeds, and it gives us the adjustment rate. So how many more seeds do we need to buy for each of these species to compensate for that? And we come out with this quantity and updated cost now of about $3 million. And we have to add to this also the cost of testing, because testing, as you just find out, is not a quick process. It takes time, getting the X-ray, interpret the image, doing the separation. So let's say it's going to be about $200 for each of these pieces.
Thank you. Afternoon, everyone. I'm Tristan Campbell. I'm a member of the CMSR group, and I actually come from a spatial and data analytics background. So not biology, nothing like that at all just data and numbers pretty much. And so I've been working on this process in the last little while. So yes, using image analysis of X-rays with X-ray data to go through and count seeds. So you put them on a tray, you haven't got to count out fused replicates, you just put them on the seeds, work out the edges, and then go through, and once it's counted them and worked out the intensity, so looking at your bright seeds versus your dark seeds, and actually classify what is viable versus non-viable seeds. And so at the bottom here, this page has got 115 seeds, 95 viable, 20 non-viable. And so this takes, you can see, particularly for some of the smaller species, this process is very manual, very laborious, and it is prone to human error as well. This one, you basically put your seeds on X-ray tray, separate them a bit, and it goes through and does it in about 5 minutes. So this is a fairly recent program working on the CSMR. It's been a very solid beta test, and so it's been used in the lab. And we're looking at ways to make this a more commercially robust and reliable species. There's opportunities for looking at things like artificial intelligence classification for different genuses as well. So this is certainly a useful tool now, and there's a lot of scope to have this incorporated in a more commercial and more advanced way moving forward as well. Unless you prefer counting up manual seeds and calculating stuff.
All right, thank you, Tristan. So that's one of the little bit of technology we've been working on with Tristan. And Tristan, you didn't tell him about the other cool things that you can do if you try to develop this. It's instead of weighing your seed, especially on some of the big seeds like of the acacia and whatnot, it might be even possible by knowing the pixel intensity to measure the weight of a single seed. So you don't need to put your seeds on a scale, but if you calibrate it correctly, you just do it all here. Put your seeds in there, count the number of seeds, and you can do like 500 seeds at a time. So it gives you already a much better number than the one we'll be obtaining now. It tells you the viability, but also can calculate the weight and the 1,000 seed weight all in one step. So how much do you think it'll cost? 20? Can you do it for 20? Yeah, okay. So let's adjust the cost. Instead of $200 per batch for the seed testing, we'll do it for $20. It doesn't change much in this total because it's just 8 species and we consider 8 batches. But if you consider a larger project, when you go 50 or 100 species, that cost really affects, especially if you have to collect many different years from different populations, getting from different origin, you see how different they can be. You'll need to test every single batch to be sure what you've got. So manage to drop down the price of the processing, of the testing, will allow to do the testing in a much more cost-effective way. So now let's run the simulation. So adjusting the price of seed, adding the operation costs, we got about 3.2 millions. With the results, we now finally got 88 million plants established, 88%. And the cost of plants went up a bit to $37 per 1,000 established plant. That's because we probably, well, we adjusted, we were buying much more expensive species compared to others that were cheaper to adjust them. But we're getting closer to our goal, which is the 100% success rate. But our success index for the diversity is still fairly low. We're about 47%. So let's look again how the species are doing. Okay, we can see eucalyptus and melaleuca are now doing fairly well. Podotheca is doing more than well. But we look at Artiplex, acacia, and Boronia. They're still doing pretty bad. And if we look at the total number of pure live seed used, so we stop counting the seeds, but we talk about pure live seeds because that's what matters. And we see the success range, we see that we are about 8% success. So every time you put a seed on the ground, you got 8% chance that that one will come up as a plant. So we said this group here, Lotus, Boronia, Artiplex, and acacia are doing pretty bad. And this now raises the questions. Are we sure that those pure live seeds are germinating? They are alive, we know that, but they're not coming up. So what's going on? And I'll have Shane Turner to talk to you more about why they're not germinating.
Okay, thank you very much for that, Simon. I'm going to talk to you about seed dormancy. So, oh, that worked well. I'm not very good with technology. Okay. What is seed dormancy? Seed dormancy is basically the inability of a seed to germinate when provided with all the necessary conditions to promote the germination process. And I'm talking about giving it moisture, the right germination temperature, appropriate light regime, and maybe a germination stimulant like a nitrate or smoke or ethylene or something like that. Seed dormancy is actually really common. About 70% of Australian species would have some form of seed dormancy present. And we recognise at the moment broadly 5 different classes of seed dormancy. But the 2 that I'm going to talk about today really or touch upon are physical seed dormancy, which is due to a water impermeable barrier, and physiological dormancy, which is basically a mechanism within the seed itself that suppresses the first parts of the germination process. Seed dormancy is mainly found in geosporous taxa, such as grasses and legumes, and geosporous are things that release their seed upon a maturity and form a soil seed bank. In comparison, bradysporous species, which we find in banksias and gum trees and Alocasiarinas, typically retain the seeds within follicles on the plant itself and only release these after some sort of disturbance. And these typically are non-dormant as such, so they germinate quite easily most of the time. And seed dormancy is typically found more often in extreme environments, such as arid, semi-arid environments or Mediterranean environments as well. And it seems to be more prevalent in understory smaller species too. So we can actually classify seed dormancy using a range of seed and embryo attributes. And the reason we do that is that we can, once we understand the type of dormancy that's present, we can actually target that dormancy with specific dormancy alleviation treatments as such. And the traits that we use to classify dormancy include things such as how quickly the seeds germinate, really, and the extent they germinate too. So we typically have an arbitrary cutoff of about 28 days. So if seeds germinate within 28 days to 70+%, we would say they're non-dormant. But if seeds take longer than 28 days to germinate or germinate to a very low percentage, we'd say that there's some form of dormancy going on. We also look at water permeability, and a simple way to do that is basically take our seeds, put them onto a moistened substrate, and then over 1, 2, 3 days, just remove them and weigh them regularly as such. And what we're doing is seeing if the seeds increase in mass or not. If the seeds increase in mass, we assume that the water's getting to the seed. If the seeds don't increase in mass, we would assume that there's a water impermeable barrier present in the seed and they have physical seed dormancy. And we can also look at the internal structure of the seed too, looking at the embryo and the structure of the embryo too. And that gives us an indication whether it has an underdeveloped embryo perhaps and requires some sort of maturation process before they're capable of germinating. So for our demonstration species, what we find is that 3 of them we would classify as non-dormant, and the other 5 we would say there's something going on with them. In terms of water permeability, we would say 7 out of the 8 water can get into the seed, but we have 1 species, which is the Acacia acuminata, which will not absorb water. So we would conclude that it has a physical seed dormancy. All 8 of our species have large developed embryos, so we don't have an embryo developmental problem or a maturation problem to deal with. And at the end of all of that, we would conclude that probably 4 of our species have physiological dormancy as such. So going back to physical seed dormancy, as I said earlier, it's basically due to a water impermeable barrier in the seed, and a couple of easy ways to actually overcome that is through the use of hot water or scarification or acid treatment as well too. Hot water is probably the go-to choice for most species with physical seed dormancy, and it works surprisingly well across a broad range of them. But having said that, it is also ineffective for quite a few species with physical seed dormancy, and in fact it will actually kill some as well too. So a good example of a species that has 100% of the seeds with the physical seed dormancy is the Australian boab. So if you look at the right-hand side there, you'll find that we get about 80% germination with precision nicking of the seeds. But if we actually treat the seeds with hot water for 1, 2.5 or 5 minutes, we get less than 20% germination. The remainder of the seeds are either not losing physical dormancy or they're actually being killed by the hot water treatment as such. So precision nicking is a better way to go for this particular species, but we've also found that acid treatment for several hours is a good way to get them to germinate too. But for us, for our demonstration species, we're lucky because the hot water works really well with the Acacia acuminata seeds. So just 2 minutes in hot water for the acacia lifts germination for about 3% in our control seeds, up to about 77% in our hot water treated seeds. Now, turning our attention to physiological seed dormancy. Now, as I mentioned earlier on, 4 of our species have physiological seed dormancy, and in fact, it's the most common type of dormancy that we find both worldwide and in Australia. Unlike physical seed dormancy, water uptake's not a limiting factor for these particular species. Water gets into the seed at all, but they have a metabolic block that essentially stops the embryo from the first prevents the embryo from developing and beginning the radical development as such. These types of seeds also have fully formed embryos as well too. And once you've actually broken physiological dormancy, they will germinate quite rapidly as such. And we just have a time series images here of Petunia longnifolia over about a week or so, where we actually broke physiological dormancy prior to the germination of them. And once they were ready to go, they grow quite rapidly. Physiological dormancy is commonly found in the daisy family, grasses and the chenopods, but it's found in many other groups as well too. And treatments to overcome physiological dormancy include after ripening, which is basically warm, dry storage for maybe a couple of months to perhaps a couple of years. Dry heat works well for some species, and that would be, exposure to 80 to 120 degrees for up to 10 or 20 minutes. Stratification, where you incubate the seeds under moist conditions at either a cooler or a warmer temperature for several weeks and then move them to another temperature to complete the germination process. Wet-dry cycles works quite well for some species such as Herbaceae and Petunia. Question?
Sorry, Shane, it's not obvious to me what physiological dormancy actually is. I know we were talking in detail, I know you're in hate, but it's still not obvious to me what it is.
The best way to think of it, it's like what happens to a deciduous tree during winter when it loses its leaves and the buds sort of retract to a structure, if you like, that is asleep essentially. And it's only through a couple of weeks or a couple of months for those buds to be exposed to cold temperatures that sort of give them the trigger that winter's passing, and once the temperatures warm up, they'll start growing again. So physiological dormancy is working in a similar way to that. Does that make sense, yeah?
Yeah, not really. No. It's still not clear. So is it a time thing? When your analogy is that it's a seasonal factor?
Yeah.
What is the big factor that is causing this particular type of dormancy?
It's basically a way for the seeds to over summer, perhaps in our part of the world. So it's a way for them to the sense that it's summertime, so they're looking for warm, dry conditions. And after so many hours of warm, dry conditions, when the temperatures drop and the moisture hits, they know or feel safer, if you like, to start germination and such. So going back to this, wet-dry cycles works for some species, and we can circumvent physiological dormancy to some degree by nicking seeds or GA treatment or manual extraction of seeds from florets. In fact, we're going to talk a bit about florets, because 3 of our species with physiological dormancy are actually dispersed within florets rather than naked seeds. In this particular case, the seeds are actually sealed within the florets as such until physiological dormancy is overcome. And a good example, that's the Triodea, where you've got the florets here. You can see what the intact floret looks like, but when you dissect it, you can see how the seed sits inside the floret, and then you can take the seed out. That's where everything happens as such. We can take the seeds out of the florets, and a lot of the time they will germinate far more easier than if you leave them in the florets as such, and that's certainly one good way to overcome physiological dormancy. But we can also acid treat the florets too, which seems to weaken the floret, and that can certainly improve germination from the floret as well. So for our 3 species with florets that we've got as our demonstration species, for the Atriplex we go from about 9% germination in our untreated florets or seeds up to about 91% after the seeds have been removed by rubbing on a rubber mat, which is essentially liberating them from the floret structure. Likewise with the Cymbopogon, we find that the germination in our untreated seeds goes from about 64% up to about 7%, again using the rubber mat to liberate the seeds. And for our Thai lotus, we go from about 34% in our control seeds / florets up to about 78% with the use of a rubber mat. For the Thai lotus and the Atriplex, we did find an improvement of sorts with the use of acid for 5 minutes, but it wasn't as good as the manual extraction using the rubber mat. So going back to our demonstration species, we basically go from before dormancy alleviation for the 4 species, and we had an average of about 51% germination as such. but when we've used the dormancy alleviation treatments for our physically dormant species and for 3 of our physiologically dormant species, we've lifted germination to about 81% on average as such. The one that I haven't touched on is the Boronia that we've got in our mix here, and I'm going to leave it to Michael now to explain what's going on with the Boronia as such.
Thanks, Shane. So I'm Michael. I'm a seed biologist doing my PhD at Curtin University with the CMSR. And I've squarely positioned myself or found an interest in our difficult to germinate flora. So over the last 3 odd years, I've been updating some work conducted by my supervisors, trolling through the scientific literature, looking for physiologically dormant species that don't have germination confounded by viability, that form a barrier to current restoration, so our really difficult to germinate species. What we've come up with is a list of around 40 plant families containing a range of species with difficult to establish seeds. Not everything on this list is difficult, but from initial estimates, we're looking anywhere between 20 and 30% of species that form a hard block on biodiversity in restoration, as we put it. Things that from a seed dormancy point of view, we don't think we're going to be able to establish with any kind of success. Looking at that literature over the last 15 years, a lot of work spearheaded by people in this very room, has given a number of families where certain things have worked. Many treatments that Shane touched on previously, things like after ripening and warm stratification, have shown to have some great success in many of these families. So we looked at the Rutaceae, and this is the group that our Boronia sit within, trying to apply some of these treatments and see what insights we can gain and how we can actually get some of these species into nurseries and into restoration sites. So we took a broad suite of Rutaceae species and we applied warm stratification and cold stratification. So this experiment here ran over 14 weeks. We had germination at a broad range of conditions, so 10 degrees, 20 degrees, and 30 degrees. For 6 weeks, we also had the shift between, so 10 degrees to 20 degrees as a cold to a warm, and then 30 degrees to 20 degrees as a warm to a cold. So that move into our winter months, which is our germination period. What we find is that a lot of things don't respond to this, but 2 groups in particular respond quite well. And this is just a very brief example of how revisiting some of these really difficult species and applying these broad suite of treatments can actually tick some of these boxes and help us increase biodiversity in restoration sites. These 2 here. But the Boronia simosa that we're concerned with today really hasn't worked here. So how can we go about increasing this, and what kind of research needs to be done to facilitate the return of this species? So nature is the best teacher, and these things do go in the wild. The problem that we face is how do we manipulate and maintain natural conditions that we can then apply to seeds to get them to go? So here we've got an example of some punnet work that we've done, and this is expanded into a greater quantity of work that I don't have time to touch on today. What we've done is put seeds into their environment. On the left, we have soil-stored seeds, or seeds that have been put out at the beginning of summer and just left there. Treatments of smoke, treatments of heat, things intimately linking them to fire, and we see that we get a bit of a result. What we do find is that when we put these things in incubators, dry after ripening, for a period of months, before sowing them into the environment during the germination season, so May, which is time 0 on this graph in both cases, we see a very significant increase in germination. That's just from putting them, over-summering them in incubators instead of in the field. This has given us a lot of insights into what's actually driving these systems and how we can begin to manipulate them and establish some of these things, not only in incubators, but also in the field. I don't have time to touch on it today, but if you are interested in some of this stuff, this work has facilitated a lot more insights that we're looking at very currently that's allowing us to achieve 50–60% germination in some of these more intractable, difficult Rutaceae. I'll leave it there and hand back over to Simon.
Thank you, Michael, and thank you, Shane. I think now, Tiana, it's your turn. So now we're going to do something a bit different. We're going to show you some of the tools we've got here and how we're going to do the processing. So I'll ask Zoe to come here and help us. What we're going to do, Tiana's going to show the first step, and then we're going to switch on to the microscope, and you'll see the microscope on the screen, and you'll see how each step of the processing will improve the quality of the seed. And unfortunately, especially the one on the back cannot see how this operates, We're going to have a break soon, so if you want to come up and have a look at this machine, you can do that at the break or at the end of the workshop.
Okay, so this part of the processing, we're doing sieving and air separation. Sieving separates any non-seed material from the seed. This way you get a higher quality batch of seeds. So if you just want to demonstrate for us with Eucalyptus leptopoda, it can sometimes be a little bit tricky to tell what is the seed and what isn't. If we know what the seed size is, essentially we know what the size of the seeds are. We can work out the minimum and maximum sieve sizes. When we pour in the seeds this way, we can actually give it a little shake, take out the different fractions. We already know that everything in the top and the bottom is waste. We're just going to, if you want to pop that in there for me, we'll have a look at this under the microscope. I'll show you first of all seeds that haven't been sieved.
It's not liking this.
Maybe in a moment.
Nothing ever works when you need it to work.
Oh.
Ah, no, I know what it is. It's Norman. This gets it off, yeah.
Okay.
Here we go.
So if we have a look at it, this here is the seed, and there's a lot of other material surrounding it that is not seed. We don't want that. So if we sieve it, just a small pinch. We're getting a lot more seeds just from a very, very quick and simple process. But there's still a lot of material there that we don't want. So we go to the next stage, which is air separation. With air separation, we're simply using airflow to carry up any material that's lighter than the seed. So all the seed remains down the bottom. If you have materials such as rocks, which are heavier than the seed, what you can do is actually increase the air separation, bring the seed right up to the top, and the rocks will stay down the bottom.
Just checking the top's on so it doesn't fly up.
We've done that a few too many times.
Yeah. So all the heavier stuff stays at the bottom generally, because I'm allowing airflow in through this side, and the lighter stuff will come up, which is generally not the filled seed. Oh yeah, thank you. So if you didn't catch that, all the lighter stuff falls up, and so you will even see seed pop up, but generally it's not filled if it goes to the top. So we like to see all the stuff in the bottom, yeah.
So if we have a look at this now, we've got a really nice clean batch of seeds. There are still some non-seed material in there, but that's okay. At least we have increased the quality of the seed batch. If we want to look at the material at the top, I'll just take a pinch. You can see that there is still material there that looks a little bit like seed, but as Zoe said before, if you were to actually look at this through an X-ray, you'll see that most of the seeds in there are not filled. They're non-viable seeds. Of course.
Where does this fit into the process? So do you do this first and then do the X-ray?
So what you would normally do is you would do the seed quality testing. You would do that with just a small sample. This, on the other hand, can be done for whole batches of seeds. So you might not get to the same purity level, but you will still get a higher quality seed. And then once you've run your batches through, you can do another set of sampling to then see what the quality is and label it accordingly.
And also, if I can add on to this, now we run the quality test whenever we got a seed batch. Even though if it's dirty and we know we could clean it, we still want to get information how dirty it is on average, like to understand it. But if you're looking more from a commercial seed producers, you wouldn't even do it. You just go straight into the cleaning, do the processing well, and then you do the quality testing. It's not point doing both sides, because it's important for you at the end is the quality of the batch. So for us, from our research point of view, we do the quality many times, but as a producer, you just do it just when you got the final product to sell.
Simon? So this equipment is fine in a lab setting because we're just using very small amounts of seed. But if you want to scale up quite a lot, there is agricultural equipment that already exists that can be used to do this on a really large scale. So if you have 20, 30 kilograms worth of seed or more even, you can run it within the space of a few minutes and get really high quality seed with ease. So next we're going to talk about seed extraction. This ties into what Shane was talking about breaking seed dormancy. Seed extraction, either using threshing, which I'll show you a video about now, or using acid digestion, helps to remove the floret, and that breaks the physiological dormancy. So this can be done on a very small scale, such as in the lab when we're using a sample. We'd use rubber mats, and a couple of the tables here have those. Or you can use the FTA thresher. Now, the tables with the rubber mats, I believe your table 4, 5, 6, 7, and 8, or maybe just 5, 6, 7, and 8, you all have seeds with physiological dormancy. So I want you to take a little bit of the seed onto the mat and try and thresh it yourselves.
So it's the people with the artiplex, with the podotheca, and the syngopogon.
And
Those,
Podotheca chrysantha.
and
Podotheca.
and podotheca as well, but it's Kingsley.
Okay, so while one or two of you are doing that, I'd like to show you this video, and we'll see how long it takes you to thresh your seeds compared to using an FTA thresher. FTA thresher.
FTA is the field trial area.
Oh, sorry.
It's our internal way to call it. But that's commercial thresher. This is a small scale lab thresher. You see it's got these rubber bits inside that thresh the seed at high velocity. Absolutely, yeah. It's automatic, so you see how it reduce and extract the seeds. Then you run them through an air separation as you see it already. There's machines that actually combine the two at once. It makes it much faster. Once they've been extracted, you can just air separation. And we see in this example, we started from 150 grams, and we end up with all this kind of external rubbish and about 18 grams of seed. This for species of artiplex. Not this one, a different one, but more or less that's how it works. And this is still a small machine. You can scale it up much bigger with larger thresher.
Okay. So you guys over there with the rubber mats, how did you go? Still going? You can stop there. You don't have to keep going. We just wanted to demonstrate how much easier it is to use large-scale machinery for this. I think they're still going. Yes?
Sorry?
She wants to know what the name of the machine is.
I'll show you tomorrow. It's
It's a thresher.
they call
Thresher.
Yeah. It's many kinds. This is a small scale one. I forgot the brand of it. It was written on it, but yeah, that's many different.
I think.
Eh? Yes, the Kimseed also have one, but this one is Haldrup. I think this one is from Europe. The one we've got here.
Perfect.
Just go ahead.
All right. So carrying on, we can also use acid digestion. This is really good for florets with a lot of chaff. It's very simple, basically just placing the seeds in a small solution, usually of sulfuric acid, letting it digest, and then using a basic solution to neutralize, rinse, and then dry the seeds. We did want to give you a little bit of acid to play around with today, but apparently health and safety aren't too happy about that, so we have a video for you instead.
So in this video you can see that a very tiny amount of acid, about 5 milliliters, was enough to kind of go and melt this half liter of Atriplex seed. It doesn't extract the seeds completely, but it weakens the external structure to a point that is much easier to thresh it. So once we run it through the thresher, for some species, you might actually get some damage, or it's very hard to extract it. But if they've gone through acid beforehand, it's much faster and you don't have damage to seeds.
So we did some germination trials. These are the same species that Shane showed you before. Atriplex amnicola, you can see that acid was slightly better than the control, which had less than 10% germination success. Acid and rubber came up to 88%, whereas rubber by itself, threshing, was over 90% successful. The reason for this is because Atriplex amnicola has really dense, hard floret. Acid itself isn't enough to break it down. You have to manually remove that floret. Tylotus polystachys, we got the best result from acid and rubber, but as Simon was saying before, not all of the species can be threshed easily. So you think the rubber's really close to acid and rubber. Why would you go to the extra lengths and extra expense of adding acid? The reason is that the Tylotus polystachys is a very delicate seed. If you just manually thresh, and you're using a thresher that's maybe a little bit tougher than basically a hand threshing that we did, it can break the seed. So using acid softens that outer coating so that we can do very gentle threshing and increase the survival of the seed. This is Cymbopogon obtectus. Acid kills this seed. So it's not a one-size-fits-all solution. You need to get to know the seed first, understand how these treatments work. In this case, we've already done the work for you, so you know you shouldn't use acid, just thresh it instead. Okay. And Simon's going to run the numbers for you again.
So the last example of a Cymbopogon was a bit of a surprise for me because I've done my PhD on grass species as Cymbopogon. And I've actually developed a protocol to use acid to clean other species of grasses, and it worked like a treat. So I was expecting to get great results, but then on that species I tried and it killed all the seeds. So you see, you cannot never make any guesses because they do behave very differently, even in the same group. Probably just went to the embryo and killed it. It's acid after all, so it just melts things. We might try maybe lower concentration, but because the threshing is working so well, what's the point of going into acid? It's when you got problem with the threshing that you must explore acid, but you shouldn't just do acid for the sake of it. It's just to solve a problem that you bring it in. Acid usually works. We've tried a few different kinds of acid, acetic acid, sulfonic acid, but sulfuric works the best. We've tried enzymes, but they're just not powerful enough. Enzymes might work well like on what's the name of the Atriplex amibacata or other species that got berries. Then with enzymes, you might be able to digest them better. But acid usually is the most cost-effective and quick way to clean them. Not all of them, some of them. Okay, let's continue our simulations, and let's see now, talk about a bit the cost of this processing. So we now found out like all sort of processing here, the sorting, hot water, extracting pure sorting, smoke, and so on. And we estimate they will cost about $10 to $30 per kilo, depending on the kind of processing we're doing. As I say, I completely made up this number. They might be terribly wrong, but it's just for the simulation. Assuming that you've got the big scale equipment and you can do it properly, not manually. And so we multiply the processing cost by the number of kilos that we'll be doing it on, and we got a total about $125,000. Kingsley, please. So we got $125,000 total. There's about 4% of the total cost of the seeds. And some might say, rightly so, why should we spend all this money to clean the seeds? After all, they're already viable. Maybe some from the Archiplex, Boronian, Terlotus, you want to do it. But why are you doing it on eucalyptus? Why are you doing on Melaleuca? What's the point to clean them up? And that brings in my next important step of the supply chain, which is seed storage. Seed storage is a super important part of the supply chain, especially if you're not doing the restoration. This very same here, you collect the seeds, you need to keep them appropriately stored. Not all seeds can be stored. Some are orthodox, it means you can dry them and you can keep them in store for a few years. Others are called recalcitrants. It means if you try to dry them, you kill them, and those ones cannot be stored very well. Fortunately, all of the species are known to be most likely orthodox, so there's not going to be many problems to store them. But I want to show you why it's important to store them correctly. This is a graph that was created by testing an European species and its behavior in storage. I know right now most people are focusing on keeping the seed in a cool environment. Keep it cool and they'll be fine. But not that really, because if you see here on the red line, keeping at 0 degrees but a 50% relative humidity, after 2 years, you've already lost half of your viability. What's the key here is probably the humidity. Humidity, if you see the line above, it's 15 degrees at 15 relative humidity. That's where you can maintain your viability the highest. So let's say we want to store our seed for 2 years. If we do it properly at 15% RH, it's a 99% survival. If you do it at 50% RH, it's going to be a 50% survival. This is an extreme example, but on many species we don't know what's the storage behavior. So we might actually losing even more than this. We probably need some more study to do it. But it's very important to get it right. Just have the right condition, because otherwise you might be losing lots of seeds and their viability. So the question is it worth building or renting a facility, humidity control, to store your seeds? Well, let's do a business case for it. We've got our, our seeds, total cost of $3 million, and after 2 years, they've lost 50% of their viability. It means that we lost $1.5 million of seed, which accounts for an average of $2,000 a day. So if your storage unit does cost less than $2,000 a day, probably it's worth considering that as an option instead of wasting all these seeds. But it's not just about that. Like we've talked about the processing. Now I want to see why processing is important for storage, because you build your storage unit, and every square meter, every shelf in this storage unit just got a cost, because you have to keep it dried, you have to keep it cool. So volumes matter. And if you look at the example of Archiplex and Nicola, that's what it looks like when it's unprocessed, and that's what it looks like when it's cleaned. This is the density. When it's clean, it's much more dense. So it's about 75 kilograms, 0.75 kilograms per liter compared to 0.2. But let's look at the total weight of it. We've got 650 kilo for the amount of seeds that we need for this simulation. Once it's cleaned, exactly the same number of pure life seeds, it's 25 kilos. Because it's not just the density, it's also the, remember, 90% of those florets are empty. So you'll be storing empty chaff. Let's put it in volume. So let's get the density divided by the weight, and we finally get the volume. For the florets, for the pericarps, we've got 3,000 liters. For the clean seeds, 35 liters. The first one, you'll fill all the internal space of a compact car. The second ones will fit in a backpack. For the first one, you'll need a garage. The second one, a shelf is enough. So that's another step why processing is so important, because it helps you to optimize and cut down the cost for storage. And storage, I cannot stress enough how important it is for the viability of seed.
Have you done that experiment where you do the interaction between treatment and storage? So whether treatment impacts viability in storage?
I haven't directly. We didn't have the time to do it because you need to. There's experiments where you do rapid aging, that you simulate condition of storage. I've been working on this for a year, so we just don't have the time. But there's lots of publication testing this, not necessarily on Australian species. But one very interesting one is in America. They've got this Asteraceae, a daisy, Artemisia tridentata. It's very important for them. It's one of the keystone species in the rangeland restoration. And they've tested to store, it's similar to the mutaceae we just cleaned. It's very tiny seeds, and it's got lots of other empty seed or chaff around it. And they've found that if you store them, even in dry condition, the clean seeds and the non-clean seeds will lose viability faster. They don't know exactly why. Probably has to do with the fact that the other material acts as moisture sinks. So kind of try to get some humidity and make the storage less effective and decrease the viability. But there's data already showing that even the lower purity of seeds, lower clean seeds, will actually last less in storage for those reasons. So there is some study out there. We haven't done it ourselves, as I said, we didn't have the time to do it. But yeah, there's so much research and science to be done here to understand the storage behavior there. It's fascinating. So moving on, it's an example for all the species we've got here, how actually cleaning them reduce the volume for each and every one of them. But as I said earlier, not all seeds can be stored. Some of them are recalcitrant. An example is Zamia. It's a very emblematic species, especially here in the Perth region. Seeds are very, very big compared to the seeds you've been working on. They usually come in this red, fleshy fruit. Once you remove the fruit, they are encapsulated within this sort of very hard shell. And if you look inside, that's what an actual seed looks like. And it's gigantic compared to all of this stuff. But it doesn't like desiccation at all. So this is a picture of seeds taken through the X-ray after they were collected the same year. First year after storage, the second year after storage. You see that over time, the seeds start shrinking and then they start cracking. That's what it looked like. That's typical signs of a species being recalcitrant. So Zamia cannot be stored effectively, and it's a huge seed, so it'll take up a big part of your storage. So our suggestion is like, don't collect them unless you want to use them, because they just cannot survive storage. Shane's got way more information about this species, got some wonderful study on it, so if you're interested, be in touch with him, because he can tell you much more about this. So we've talked about sim processing, and now we'll be going into the simulation. To wrap it all up, we went through the cost of the processing, but before we go into the simulation, let's have a 5 to 10 minute break. You can come up here, have a look at the equipment, go in the back of the room, have a tea, and we'll start again in about 10 minutes. Any news from the...
Off this laptop.
Yeah, go for it.
That must be cool. We'll get some.
Yeah.
Just plug my phone into charge.
Yes, sure.
Does that have a... Paul will be back. That doesn't do Paul's coming back. So the guy that came in late is Neil from Woodside.
Is the guy talking to Kingsley?
Okay. I was like, Kingsley, look at his...
The magic mic.
Curious to ask a bit more, because you had the Kim seed zigzag,
Yeah.
but as a machine, even though it's got its teething problems, really.
Run away from it. It's a piece of rubbish.
Yeah, no.
Don't waste the point on
looking
that.
at- There's one in from Holland, the selector.
Selector, that's much better.
Yeah.
That's worth it.
But in terms of like a column separator versus a zigzag, do you have like the same sort of control that do it a much more efficient?
Yes, it's size and efficiency. Yes. I was about to buy a selector, but then the Kim seed was much cheaper, and I said, oh, it's going to be the same. Let's get a complete piece of rubbish. Like poor engineering, poorly built, really bad for, like it might work well if you're cleaning, I don't know, canola. But if you get the fine tuning, the selector might be double the price, but it's worth every single dollar.
Yeah, okay.
And they are not the only one. I've got seed processing Netherlands. They do similar machines. But my experience is mostly with the selector ones. Now they are out of the...
Right.
And they've got like a commercial agency. That's interesting.
Yeah. Right. Is that the selection?
now the building.
Three different models there.
Yeah, and I think the bigger ones.
Yeah.
But those.
Yeah. Because we've got two of the columns separated. So it's just whether or not.
That's
Like,
it.
can you basically just make match in?
One because it's got vibrating network, especially if it's large, you can just start to 5.
Yep. Making things, yeah. And then you've also got the papery. Yeah, but it's papery.
Exactly. So you will need the one and so you can just run the wings and sides it. And then you can run it again if you want to separate.
Yeah,
So the column
so you've got
separate.
it, so you've still got the fine control back here, but I guess zigzag itself doesn't like the stuff that is mentioned.
Exactly. It makes it a bit better.
Yeah.
And it's easy to regulates the intensity, and another mode regulates the speed of the vibration.
Yeah, okay.
Once you go manually to open it, basically takes forever.
Yeah.
Seats pop over they operate because they haven't worked out their complete waste of yeah, poorly. Right? There's some ideas that like I fix it in my life. Thinking about
yeah, well, I used to work for yeah, I started off working for Kingsley.
Some of the machines I got them the clippers.
Yeah. They're pretty good.
Yeah, I still have to buy, but those are like the clippers just vibrate. So
We've got a wooden version of that.
the old clip.
Yeah. Yeah. But we don't even use that very much. I just do it with hand screen.
No one necessarily like it.
Yeah.
All the hand collections you can use it around the It's more like if you run it.
Yeah.
It's got kilos and kilos. That's something like that.
Yeah.
Calibrating the well. But yeah, come to me if you want any questions. Because I actually met the guy that Haley? 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 Can you ask them all to come back? Well, you point by hand and then I'll tell them on the microphone. Come on, start again.
Okay.
If you could please go back to your seat, we're about to start again.
Oh, that's nice to hear.
What slide number are you up to roughly? That's 70?
I've got it.
Okay.
Because now it's just me, so I can speak. Well, if you'd like to. If no one wants, probably doesn't. Do you want him tomorrow tour? Do you want him tomorrow the lab tour?
Yeah, I'm not sure. I don't know what I don't know. I haven't been. So honestly, I don't know. If you want to come along.
Yeah, absolutely. So show around the X-ray a bit better. If you could please return to your seat. We're running out of time. Okay, I hope you enjoyed your coffee and your break. So now we might continue with the talk. I've lost the... here we go. Okay, so we left before with the cost of processing being about 4% of the total. And now we can finally run the simulation with those numbers. So we know that the total cost, including the quality testing, including processing, is $3,200,000. So this is total seed cost of seeding operation, and total cost goes up to 3.4 million. We run now the simulation, and we get, finally, 110 million plants. So we've exceeded our actual goal, which is now 110%, and the cost for 1,000 established plant is now $31. Our index is, however, a bit low because we've shot too much and we've got too many species compared to what our target was. So if you look how each species is doing, we see some of them are doing much better, like some of the eucalyptus, podofica, and so on, but others are doing worse. Boronia, for example, is not doing very well at all. It's just 400,000 and we need 2 million. So if you look at the total number of pure life seed used and the success rate, on average is 14%, but Boronia is just 2.4%, so it's not doing very well. if we now look at the cost for 1,000 plants for each of these species, then we see that the cost for Boronia is all the way up to more than $700. And that's because the seeds are so expensive and they're not doing very well. A way around this, it's probably changing this number, because as we've seen, Michael showed us, Boronia is very hard to work with, and it still requires lots of study to understand how to germinate it more effectively. So instead of keep on throwing seeds out there for them not to germinate or do very little, and spending lots of money for it, why don't we just reduce that number of plants? Our customer will understand if we justify well enough, and increase maybe the other numbers of the shrubs so we can reach that number of shrubs. And maybe instead of doing seeding for that species, we could use a seedling instead. So we might try to get some establishment with seedlings, or maybe invest in more research and understand how to make them germinate, instead of just throwing seeds out there and just wasting our money. So if we now correct the total target with these numbers, and we adjust the total numbers, we got a cheaper price. We're saving about $200,000, adding the operation cost $3,200,000, and we finally get 100 million plants, which is a 100% success rate, cost per 1,000 established plants at about $33, and a success of 99.8%. So we did it. Finally, we managed to get there and to get our targets. Every simulation from now on, what we'll try to do is to make it more cost effective. So decrease the total cost and decrease the cost of 1,000 established plants. But we're good. We have reached our target, so that's a great result. Okay, before we move on to the next simulation, I want to open a bit of a parenthesis on now we sell seeds and how we consider the cost of seeds. Because have you seen so far all the processing, the seed testing? This is all quite expensive operation. But what is the interest of a seed supplier to do all of this if what a customer wants, all he wants is the cheapest price per kilo? If they don't care about purity, viability, they don't care about the fact that they'll germinate or not, why a supplier should do this? A problem is because we keep on talking about native seeds in kilos or grams or whatever. We use the price per a quantity, per a weight. I think we should move away from this concept altogether. In my work on agricultural species, because it's kind of guaranteed that they all germinate and you know exactly what a seed size is like. Not for natives, because kilo doesn't tell you anything about the seed size and weight, and we know how these affect the quantity of seeds. Doesn't tell you anything about the seed quality. You'll have to go and do the testing yourself. And remember, the final outcome of restoration is not how many kilos I put in the ground, it's how many plants I get at the end of my project. So my suggestion is that over time we should try to move away from the price of seeds per kilo and move instead to the price of 1,000 pure live seed. I'll show you now how it's calculated. So you've done the exercise of testing the viability and the purity, exactly the same example Tiana showed before. And we know that the viability is 72%. We know what the 1,000 pure seed unit weight is, 1,000 seed, it's 5.7 grams more or less. So we combine these two numbers, we now know what the weight of 1,000 pure live seed is, about 7.9 grams for the example. We also know the price of that batch of seeds per kilo, let's say $1,000. If we now put these two numbers together and multiply the seed weight, corrected in grams instead of kilos, but the seed price corrected in grams, multiplied by the 1,000 pure live seeds, we now have got this number, $8.6, which is the price for 1,000 pure live seed. You know exactly what you have, and when you do your restoration plan, I need to buy 10 1,000 pure live seeds to reach that goal, so you know exactly how it costs, and you know exactly how many pure live seeds you're going to put in the ground. Yes?
I would say it's much harder to organize 1,000 seeds than it is to organize a kilo.
That's because we're used to do grams. But if you start really thinking, because at the end of the day, the information that matters, it's this.
But you have to individually count it, right? Isn't that how it works?
No, it's not the counting, it doesn't matter. All these results, I can get it from the data you've done before. You know what the viability is, so you estimate what this is based on the data you've got. You don't have to count them. It's if you buy seeds, if I'm doing a restoration and I want to buy seeds, instead of buying 10 kilos of seeds without knowing how big they are and without knowing what's their quality, I'll need either to get someone to do the testing or do it myself. I'll eventually end up with this number. Why don't I buy the seeds directly with this number? It's like something that if it'll ever happen, it'll be a while because it's hard to change like an industry and how they do things. But over time, it's good that we start thinking about this one, because if then the customers start thinking, oh, that's the information I need, and ask it to the suppliers will probably start complying, because for them as well, they can get their work paid, because this number will also include the cost for processing, the cost for cleaning, the cost for giving you germination, and it will all be included in this.
It's like when you buy this public little seed packet that says 200 tomato seeds, and you know the germination rate is 99%. So basically you're buying not 1 gram of tomato seeds, you're buying 200.
Yeah, exactly, yeah. So I've tried to plot this still on my original data of 131 species, what the price for 1,000 pure life seeds would look like. On average is about $6.7, and we see how our species stack compared to these numbers. We see the grasses tend to be the more expensive, about 7.5, but all our species are more or less in line, apart from Boronia that is up there at about $17. So I'm not hosting the meeting for some reason. I would say okay. So, close parenthesis about this new way to think about it. I don't say you have to do it tomorrow. It's going to take lots of time to get people used to the concept. But I think that ultimately this is a much more useful information than just talking about the cost per kilo. So, back to our simulation, finally. How much a species are doing? We know what the success rate is. Now what we can try to do is improve that success rate. Bring that 14% up higher to get a better success. A way can be done by improving the efficiency of the seed delivery. Right now we've been using like this 2-row seeder, and we need to mix our seed because all the seeds are different shape and size. We need to mix them with lots of inert material, vermiculite, perlite, to make sure that they're evenly distributed. But what if you can use this one? We can use like a 20, 40-row canola seeder. Those are already available. They're used constantly around Western Australia. What if you can get our native seeds mix through that machine and deliver a larger scale and faster pace? There's a problem though. Our seeds are still very different and diverse. You remember the seed mass distribution? Once we clean them, all of a sudden they all become smaller. And our canola seed, the reference seeds we try to mimic, it sits within the 1 to 10 grams. It's usually between 7 to 8 grams per 1,000 seed weight. So our goal now is try to get all these small seeds from the Melaleuca to the Singaporean and so on to reach the size of a canola seed so it can go through the seeder. So moving them from here to here. And that can be done with seed enhancement technology, and especially a technology called seed coating. Seed coating as a principle is very simple. You just put stuff around a seed. When you put very little amount of stuff, it's called film coating. When it's a bit more, and you can still see the shape of the seed, it's called encrusting. When it's so much that it turns into like a spherical, a little ball, then it's called pelleting. It's been done in agriculture for almost 100 years now, and one of the main reasons why they do it is to modify and standardize the size and the weight of the seed. And that's exactly what we're after right now, especially the pelleting. So I'll show here an example. This is Atriplex nummularia. And we've been using a rotating pan. It's the equipment for pelleting we've got Curtin. So this is the original seed. This is what like the pan looks like. This is the small one. We've got a bigger one as well. So you keep on adding material, alternating dust and a liquid binder that works like a glue. You can add some color at the end, and that's what they look like. So on your table, on your species, not all the species, but most of them, you'll also have an example of what the seeds will look like after they've been pelleted. The acacia don't have it because the acacia are too big, we didn't pellet it, but the other ones do. And also you've got on your table, given a little package, this one you can take home with you. And there's an example of a pelleted seed. This is for Negoxanthus menglesii, the kangaroo paw. And we've got the untreated seed and the pelleted one, and you can actually try and plant them in your garden.
What's the life of the kangaroo paw in this mode?
Sorry?
What's the graph of the life of the seed?
Do you mean the pure life seed?
Yeah, like over 2 years. What is how does it What does the graph look like?
Does it impact
yeah.
viability, dormancy?
We don't know. The honest answer, we don't know. Like, it's so new, this technology, that we didn't even have the time to actually try it and then put it in storage. Ideally, I wouldn't do it and then store it, because you are storing lots of material. So just store your seeds as they are. Before your planting season, you start doing the pelleting, and you pellet what you're going to use the next season. Otherwise, you'll be storing a big bag of mostly clay. So it's probably better to do it straight away, and also because we don't understand how well they will store as well. So my safe bet will be pellet just before you do the seeding, or a few months before, but not years in advance.
But if you could issue around breaking dormancy,
Sorry?
you could issue around breaking dormancy, issue that do immediately growth dormancy.
Yeah. So for example, some of the, like all of the physiological dormant, well, you remove them from the casing, from the shell, they can germinate straight away. Then you put the pellet and they'll germinate very well. For some of the other stuff, you need hot water treatments or acid. You can treatment first that and then run them through the pelleting. Now we're actually working on some way maybe to do it at the same time to optimize the process. But we're still working on that. Another one for the species that need smoke, for example, now you just put them in smoke water or whatever. Instead, what if we use smoke in the binder mix? So we're actually inside the pellet, there's already the smoke particle. So when it gets imbibed, that smoke will eventually get to the seed. You're now stealing my thunder. I'm about to talk about that. Don't spoil it. So we've got here an example of how it changes the size of the seeds. This is another Melaleuca cestina from here in Perth. This is 2,000 seed, and this is still 2,000 seed after they've been pelleted. And this is an example of germination of R2plex nummularia, and usually it doesn't affect germination. I think, Hayley, you've just run a trial, and it seems like it's doing fairly similar to the normal one. Yeah. So let's see how much it costs for pelleting. We've got an estimate of more or less how much it would cost to pellet the species. Some are much more expensive than others, so especially the very small one. The Melaleucas, it takes a very long time to build them up from a super tiny seed to the size of a canola seed. And we put together the price for all the species, and it's about $170,000, which is more or less 5% of the total cost. Seeding operation. We're now moving away from a small tractor and getting a much bigger tractor with a much bigger seeder. And let's say its cost becomes $300 per hour. But the good point about having this such a large machine, you can do 10 hectares an hour instead of 2. And that brings down the time required to do all the seeding over 2,000 hectares down to 20 days from the 100 that were originally used. And that means it's actually going to be cheaper, just $60,000 to finish your entire area. So if we now run the simulation, we see cost of $3.2 million at the operation cost, and we get the result. Oh wait, that's better. We get 110%. Doesn't matter, let's adjust it, because we'll have to do the species adjustment again, so let's resimulate it with the adjustment, and the total cost is now 2.9 million. Again, add the seeding operation cost, and let's get a simulation. Well, we get 100%, because we know that's what we get. 99% success. And now the cost per 1,000 established plant has got a bit lower. It used to be 33, now it's 30. So we just managed to save $3 per 1,000 plant. You might ask, was it enough? Like, is it worth to do all this stuff if you just save $3 for 1,000 plant? And I've got an example to show why it might be very important. This is a real experiment that we ran this year in collaboration with Green in Australia. They've asked me if I could pellet some of this very small seed. So 2 eucalyptus and 5 Melaleucas. We've done all the stuff we've seen today, the quality testing, processing, the pelleting, and we've got all sorted. And then we decided to test seedling emergence in a field trial that was done in the Eastern Wheat Belt, and tried to understand what the survival of the seed would have been after seeding with manual seeding and also mechanical trying using the tractor. I've worked the number they provided me, the amount of seeds they put down, and they use like grams per hectare. And it's something like 450,000 pure life seeds per hectare they actually put in down there. And they've got an expected result between 2,000 and 5,000 plants. That means that the expected success rate for this species is about 1%. So I've adjusted my quantity for the pellet to match exactly the number of pure life seeds, so we know that we compare it like to like. And then we went out to the field and tested it. So we've done unseeding, and then we've done with the tractor over an area of about 1 hectare. I came back in November and done the scoring, and it was terrifying. So out of 6,800 pure life seeds that we put manually, we got 8 seedlings surviving. If you look at the mechanical seeding, out of an estimate of 450,000, we got 700 live seedlings. It was awful, like something went terribly wrong. Fortunately, before leaving, Michael put in the ground this probe that can help us measure the temperature, but most importantly, the water content in the soil at different depth, 1 centimeter, 5, 10, and 20. And we've got this graph. So if you look at this graph up in the right, you see that from September to November, the soil moisture content drops drastically. That's because there's been no rain for almost 2 months. So if we look closer, and if we look especially at the blue line, which is at 1 centimeter, and the red line at 5 centimeters, we see that it was bone dry for almost 2 months. But if you go a bit deeper, at about 10 to 20 centimeters, there was still moisture down there. So if the seedlings managed to grow a bit more and get access to the moisture, they would have survived. But probably they were not developed enough to get down there and get the moisture. So they all died. They emerged, but then they died. But if you compare July seeding, which is what we've done, it was the 15th of July, to something they've done previously, in June, there are lots of stuff coming. There you can see the eucalyptus, you can see the melaleucas emerging. And that's probably because they had enough time to dig deeper and to access the lower soil moisture. So the lesson that we learn from this experience is the later the seeding is in the season, the higher is the risk of mortality because your seedlings don't have enough time to grow the root and access that water. And so the message is concentrate most of the seeding early in winter from March to June, because when you push it further, you're risking to lose most of your seeds. So if you look here, our current model, like with the previous seeder, 2-row seeder, in 100 days you can do the whole thing, but that forces you to push your seeding all the way into August in a very high risk area. But if you got a canola seeder, you can concentrate all of it at the beginning of winter and give all your seedlings all the winter season and all that water to establish deeper root system. So that was a very important lesson. I didn't factor this into the simulation, but when you plant your seeding, you have to take this into consideration because the risk, if you go past July, becomes very high of you failing the entire project.
So is the point of that your experiment?
Sorry?
Is the point of this that your experiment didn't give you a good result because on seed coding because of time? So really you haven't done seed coding experiment?
No, exactly. Eventually, it tells a completely different story to what we're expecting. We want to compare like how pelleting, if it does improve viability, and what the seed emerge or not.
The actual conclusion was plant early.
Exactly. So it was a completely different thing than what we planned for. We just had to change the. Yeah. That's a good question. I've chatted a lot with the actual guys from Greening Australia driving the tractor, and I told them, why don't you do dry seeding much earlier? Once they're in the ground, they'll stay there. Once the moisture comes, then eventually germinate. Especially if they're pelleted, maybe they're less subjected to predation like natural seeds are, so they will have probably a chance to sit there. And he told me that especially in the wheat belt, wind is a big issue. So if you sow them, even if it's down like 0.5 centimeter, 1 centimeter, if you got very strong wind, and if it's very dry, you lose the whole thing. So you need the first rain, you need to get some moisture in the ground, and then you can start. So the beginning depends on when you get the first rain. So whenever you got that, then you can start. But if you get the first rain in April, that even shrinks even further your window. That's why having larger operation, larger machine really helps getting through faster. That's seeds, yeah, that's seeding. Okay, so far we just consider pelleting for its mechanical properties, so that we can make seeds bigger and we can just push them through the tractor at a larger pace. But we haven't put anything on those pellets. It's just clay. It's just the material to make them bigger. If we look how each species is doing, our percent of success hasn't changed at all. It's still at 14%. But we want to make it better. So let's see how precision seeding for a canola seeder can be improved using pelleting. we've already done as much as we could on the microsite delivery, 90%. It's a pretty good result using the precision seeder. And now many seeds will germinate at 80%. It's a pretty good result. But how can we improve the 40 and the 50% in the emergence and the survival? There's many things we can do. Something like protectants, help seeds from predation, or surfactant agents. So if you've got problems of water repellency, we can put that in the pellet and make sure we create a good microsite for the seeds to germinate and access to water. Hydrogels are another things that could be used. So let's say by doing that, we can improve the seedling emergence from 40, let's say just to 50%, just a slightly bit. And then we can put something else on top to improve survival. I've just got a paper recently published where we proved that salicylic acid, which is the natural compound that forms aspirin, helps plant survival. And we've researched this on ground species, and we showed that it does improve survival compared to the untreated control. Sorry?
Mechanism understood?
Yes, but that's a very long story. Maybe later we'll talk about it. So we can say we might try to improve the survival of another 10%. So just raising a bit the emergence and the survival, and this is the...
Can I ask a question?
Yeah.
Sorry to interrupt, but is there something in the pellet also that can suppress weeds, as well as help the seeds germinate?
That's...
Something that can stop weeds encroaching on its patch?
So that's another series of studies done also on pelleting or like added material. It's not like putting on the pellet itself, because if it's detrimental for weed, probably is also detrimental for your seeds. But it's actually put compounds that can protect the seeds from herbicide. So I think activated carbon works fairly well. Put activated carbon on your pellet, then you can kind of spray the rest. It kills the weed, but it makes your pellet, like hopefully your seeds survive. Yes, Kingsley?
Yeah, I just, good question. So we published a series of papers in the 90s on salicylic acid, and we've only just started revisiting this and putting in the papers. One of the papers was about making Bigfoot get herbicide resistance into the early stages of the seed with salicylic acid. So it's only just salicylic acid work. Now we're beginning to start a whole series of trials. And it also included frost resistance and resistance to salinity. Now, it doesn't mean they hold that resistance to salinity for life, but it means if there's a slight concentration of salt in the surface layers, they may get down a little deeper. So I think the salicylic acid story is starting another new journey. It's certainly the work that we did in the 90s. It's our most highly cited work, sits at the top of the WASA. But until we'd perfected the delivery to the seed, we had no way of putting aspirin over farmland. But we've now solved that problem.
Okay, so let's move on. Sorry, I have to go quickly. They're going to kick us out at 4, so I have to rush through my last slides. So let's say that in the simulation, we've got now pelleting with our good stuff, that being salicylic acid, that being the hydrogels, and the water repellent agents. And we see that now the total cost is much lower because we probably need less seed to get the 100%. So if we got the seed cost, it's almost $2 million. And if you look at the results, well, it's always 100%. And the cost per 1,000 established plants is going to drop down to $20 for 1,000 plant. So you see, just modifying a few of those transition phases in the life of those seeds, so the emergence and the survival, you can really increase the efficiency of your seeding. Now, let's look now what it's doing. So we move from a 14% success rate now to a 21% success rate. Still not great, but it's much better than it's ever been. And there's many more things that we can test and we can try. One was, example, try to include rhizobia and microbes. That's something else that would be very interesting to try. Some things include germination promoters like smoke. We know how to do the pelleting now. It took here to get to this stage. Now we want to move forward and try to develop new technology and new ways that we can deliver more effectively and bring it up from 20% maybe to 30%. And you see how it affects the cost. That's huge for grabs, but we really need to put some science and research to understand how it works. But let's take a completely different look. Let's move away from the pelleting and the technology and look at another way that we can make the seeding cheaper and to make especially the seed cheaper. Now, if you look at the species that we're using for the simulation, let's look at the last 3. the Amarantaceae, Poaceae, Asteraceae, the forbs, grasses and annuals. We use lots of them because they are the ones we want to cover the ground, and their cost is quite high in the total because we need lots of seed of those. And the price per kilo is also quite high. Asteraceae, for example, up to $2,000 per kilo and so on. But those species could actually be fairly simple to cultivate and to start producing in a controlled setting. It's not trees, it's not shrubs that might take years or decades to actually get the amount of seeds you need. It's important to go down that path as well, but we can start immediately growing some of these forbs, annuals, and the grasses. Try to set up a seed farming on a large scale. And doing so, you can control the irrigation, control your weed better, have mechanical harvesting, immediately process your seed. Instead of having them sitting in your car for days before you drive back to the processing, you go straight into processing, you immediately clean them, you reduce the amount of pests and the insects that can eat your seed, you can dry them straight away, and you can increase the quality of seeds, increase the viability, and ultimately decrease the price. In this graph, we show a bit what is the balance between natural collection from the wild and cultivation, the production. Usually, if you collect from the wild, it's more representative of the genetic of the original population, but you have the risk of damaging the population, and it's very expensive. But if you improve your seed production, then you can bring up the scalability and drop the price drastically. I've seen this happening time and time again in Europe and in America. You take those species that a very common one, Achillea yarrow, it's got Asteraceae with white flowers, collected from the wild, $2,000 a kilo, or euro per kilo. Produced in Germany in the farms, $80 a kilo. It's immense, the cost reduction if you do it properly in seed production. So let's say in this example, we're gonna cut by half the cost of these seeds. It probably can be more, but let's just do it by half. So instead of 320, it's gonna be 160 and so on. And now the total cost of seed is down to 1.2 million. Adding the operation cost, simulation always 100%, but now the cost for 1,000 established plant is $13. Remember, we start from 165 at the first one, and now we're down to 13. It's a tenfold increase in our efficiency in seeding. So now to wrap it all up, I want to show with this last slide a bit of all this journey we've gone through today in this workshop, with the total cost in red and the success percentage in blue. The first example using broadcasting, $260,000 with 1.6 success rate. Second example, multiplied by 30, we literally go off the chart for the cost, which was almost $6 million, and yet we couldn't go past 50%. Let's do precision seeding. The cost drops drastically. We now got down to $1.7 thousand, and the success is just slightly higher. Now we test for seed quality, cost goes up, but so does success. We start adjusting for dormancy and do the processing. Now we actually over-succeed. We go above 100%. So if we adjust for the species, we can bring down the cost a bit, and we now reach the 100%. And from now on, everything we do is just to make it cheaper. So adding the pelleting, it brings it down to 2.9 millions. And then we add pelleting with beneficial compounds, it brings it down to 1.9 million. And if we put seed farming into the equation, and so the decreased cost of seeds, we can bring it down to 1.2 million, and always having 100% success rate. Now look at the same thing, but taking in consideration the efficiency, so the cost for 1,000 plants, and the diversity index that we created. First example, $162, of a success of 0.7. Second one, $115, success 23. Then we go all the way down to 32, because we do precision seeding, success is still around 24. Cost goes up a bit when we do quality adjustment, but the success goes up a lot, all the way up to almost to 50%. Breaking dormancy and doing the processing, the cost drops to 31 and the success goes up to 75. Finally, adjusting the species number, we can go all the way up to almost 100%. The cost goes up a bit to 33. Doing the pelleting drops it to 30. Pelleting with good stuff down to 20. And then all of this plus seed production, it brings it all the way down to 13. So this is the end of the workshop. If you're interested and want to read more about what I've talked about, something is inside this series of documents. It's a series of publication. It was published last year in the journal Restoration Ecology. And in here, we break down the seed supply chain in all the different components that I talked about today. A very interesting document to read, to have an idea of what's going on in Australia right now, is the Australian Native Seed Survey Report. It was prepared by this group of people. Paul Gibson-Roy is one of the authors here, is also on the call, and if we got a chance, we don't have much time, it would have been good to have a chat with him online. And then another interesting document to have a bit of comparison to what's going on in Australia to what's happening in the United States is the National Seed Strategy. This is a document created 5 years ago by the Bureau of Land Management, and it's impressive to see the difference. Right there, right now, they're doing seed-based restoration at a scale that is unthinkable for us. Just in terms of public investment of the public agencies like BLM, Forest Service, National Park Service, in 5 years, they've committed for $500 million in this native seed market. So what are we doing in Australia? Is there anything comparable to that here? No. And we need to move towards that direction, because here we're talking about this grand statement. We want to restore the wheat belt. We want to bring back hundreds of thousands of hectares. How are we going to do it? We need this plan. We need this sort of support to be able to invest in this technology and to test it seriously and to improve the supply chain and to improve the amount of seeds we get, improve the quality. All right, that said, I'd like to thank... We got till 5? Okay, so they just told me we got the room until 5. Good, it gives a bit more chance to chat and have a bit of questions. So I'd really like to thank everybody that has contributed to make this workshop possible and helped me working and put up with me in the past year or so working on native seeds. It's not been easy, as I can testify. But yeah, thank you very much to all of them. Thank you for Kingsley to put this all together. And I think Kingsley wants to say a few things after me.
I think the word for the workshop would be exquisite. I think what you presented, I was a little anxious looking at the workbook because that's why I sat down in the back. I'm thinking, oh, I couldn't fill this out. I always flunk maths. But you're very careful and well-considered way that you've, I think, walked the audience through something that there are just vast assumptions in this new national agenda that frightens me because we are undercooking what we can do as a thinking nation. The National Seed Strategy that you saw there, so I was part of the team with the Bureau of Land Management that created that. If you look at the dates, 2015 was Obama, of course, and then Trump came into the middle of that. He cancelled the whole thing. Biden came in, as I started my intro, and has revived the whole thing and re-energised it. And it is astonishing to see the rural and regional economies of the US buying into this because they've got a belief in the system. And they're bringing back their sagebrush and they're bringing back systems right across the Great Basin, which have been so damaged through that sort of investment. And the great thing is, you know, they've probably now gone to sleep, but we've got a whole range of US people who were listening into the call because Simon and the CMSR team are part of that international group that sit over at this side. We're still making baby steps. The interesting thing is what you hear today, you might say, so where are the other native seed research groups in the country? There are bits and pieces, but none that bring it together. We have 4 weeks left in our centre, so in the next 4 weeks this is a great opportunity to pick up on what you've seen us start. What we're hoping to do is continue the life of particularly some of the major programs. So we have what we call our packaged projects, which are for industry to come back and talk to us. So just ask Vanessa, if you put your hand up, or Hayley. We have a series of package projects where we're now talking to partners and new industry and other individuals about elements of this that they think are useful. So Curtin's investment in this has been substantial. They've put altogether $2 million in cash. that to actually materialise the whole seed side of the program. So this is no small investment, because they believe this is an incredibly important part of building a robust part of what is the future of the restoration economy. Now, some of you will know that the restoration economy is part of what is now the biodiversity and conservation economy, but it also needs to be part of a very important and missing piece, which is the Indigenous empowerment economy, building rural and regional jobs through Indigenous communities. We have a major bid sitting with the Federal Australian Research Council for another centre, which is called Healing Country. For those of you that made it to the museum about 5 weeks ago, we had Healing Country, where we had Indigenous people speaking about what they could tell us about their country, and then the Western scientists joining that those concepts to say we can work Western and Indigenous science together. So the journey that we see going forward is a very exciting one. It's a one where we have to rethink that one size fits all across these vast landscapes. And we have to remember that if we don't get this right at this extraordinary pivotal point, the COVID pause has made us think about the environment and nature. The carbon economy has finally got respectability, a bit late, but better late than never. But now's the opportunity to rebuild our bushlands, our conservation, building real economies into it, getting the carbon locked away, and future generations will look on this moment in time as the time Australia did turn the corner. So look, I'd like to thank the team. I think they've done a splendid job. If you could join me in thanking them for a lot of hard work, careful planning, and it went tickety-boo. Thank you very much. So you're most welcome. There's some coffee. The tea urn's dried up, I'm afraid. I was drinking so much tea down the back, and I do apologise if I was talking during them. I think it was important discussions. But please feel free to mingle with the seed team, and we're always happy to talk, and you've got our contact details. By the way, the booklet at the front lists all the seed team, so you can get their profiles if you haven't remembered who they are. Thank you very much.
Before you go, one last thing. So we've run the workshop today. We almost had to close registration because we reached maximum capacity, even though lots of people didn't even show up. But we think that maybe if we see more interest from other groups that couldn't make it today, we might run it again on the 9th of June. So if you know someone else that might be interested or they think should listen about this, please tell them, let us know. And if we reach at least 20, 30 people, we might run this again. Maybe not for you, because I won't ask you to go through this twice, but if you know someone else, please let us know. And yes, if you got any questions, any discussion, you want to come here and look at our machine, or if you want to come and visit us in the coming weeks to Curtin, just let us know, and you're always more than welcome. Oh, any questions?
Sorry, thanks. It was a tremendous presentation. Thank you very much. We're doing 700 hectares of planting biodiverse in the Midwest this week. We had a bit of a slip up with our planters that are using fertilizer, and that we didn't use fertilizer in the previous planting, and it was pretty successful. We had a very high success rate, but now they're using fertilizer, and it's new for us, and we're not sure whether we should be doing that or not. Could we ask for your opinion, please? And you might ask what fertilizer, and Natalie knows.
I think maybe Kingsley would be better to answer that question.
Yeah, it's a really interesting one. Everyone, you get this native fertilizer. I tried a big native garden, and it was very successful in the first year. It was pretty 1,000 plants. And I thought about it, and we do a lot of nutrition work, and this is work that I do with Hans Lambers, who's an ecophysiologist. And I thought, okay, that's the proteaceae. They're the Banksias and the grevilleas and things, and they've got special roots that mine phosphorus. You put phosphorus on the top like superphosphate, you kill them. But then what we did, we drove an entire native plant fertiliser thing, which was no phosphorus. And so lots of our plants love phosphorus, but we starve them because that's what native plants do. So I've learnt the lesson. I have a whole big plant tablet. The Manutech tablets goes right under my plant. They go on the top, and it's terrific. So I wouldn't be terrifically worried, but you don't top dress any of the family that has those cluster roots, for example. And you don't overfeed, so you get overtopping and small root production, which means when summer dormancy hits, you've got the root shoot ratio is all out of kilter. So some balance.
Have you monitored the difference between the same area?
Yeah. So we've done this actually at City Beach in June restoration because we were getting constant failures in our community group, and that's where in about 2005 we started. I said, let's set up a trial where we're going to get complete tablets, not the native ones, and put them under the plants, and the difference was dramatic. So the community now do that regularly, and they use the one called Typhoon. But to me, so some very simple trials, even this growing season. We're now getting, right now is the perfect time. You saw the plot. I always say to people, look at when farmers seed, and if you seed outside of that window, you're probably going to be in a bit of trouble. If you're using native seed, for example. If you're using native plants, you get a little bit more leeway in terms of that planting time. So if you're, but our vision, and this has been the great journey with the seed team and particularly with Simon, if we can't use a canola seeder, and sure, there are Banksias and some of the other bigger seeds which will need adaptations to that, but if we can't use a canola seed, we're never going to do the 1 million hectares a year. We're never ever going to achieve it. So we've got to think like farmers, and we can bring the technology to deliver that farm-ready capability. And we've got Hayley here from CSRO, and the Chatfield people, where we've now got the first pelleting. Hope I'm not giving away all our confidential information. The first pelleted salt bush, which means the potential to direct seed salt bush, may mean that there's a bigger area more cost effectively that you can do. There will still be cutting grown salt bush and particular cultivars, but real opportunities. But unless we can put our native hyper-diverse material through a single 40-prong seeder, put 40 species or more in at a time, then you come back and do another 40, and you're doing all of those hectares, we're not going to achieve the carbon budgets, the biodiversity promise, or repatriate land that's going into great climate damage, particularly in the eastern wheat belt.
Okay, before we get another question, because we got the room until 5, I would actually like